Resume the recording. Share my screen. Well, we don't need that. We probably don't need that either. Okay, a reminder, the extra credit study sheet is available. Click on this link in Canvas. So it's near the very top. What is that? 1, 2, 3, 4, 5, 6, 7 links from the top. And then I'll tell you a little bit about the assignment, not much. And then click on this link and you'll download the spreadsheet, which you need to fill in to obtain the extra credit. It'll be worth 6 extra credit points. And all it is you're going to be planning out your study schedule. Are you guys hearing me okay? Okay. Last time, I think it was all of yesterday, I didn't have this plugged in. I thought it was plugged in, and apparently I had put my thumb drive in and I had to unplug it, and I forgot to plug it back in. Let's see. There is another extra credit assignment. I think I've mentioned it. You can send me a picture of your face. It'll go in my gradebook. No one will ever see the picture. If you don't have a digital picture, just get somebody with a phone and ask them to take a picture and email it to you, and then put it in this folder here, EC for extra credit pic for extra credit picture. And you'll have either a link over here or at the bottom of the page for downloading the picture. It's worth 3 points. The purpose of the picture is just to help me learn your names. When I see your smiling face next to my gradebook, I will associate your face with your name. The lockdown browser requirement is now working, so you can download Respondus Lockdown Browser on your system, and you will need it to take the quizzes and the final. Please get it downloaded before it's time to take the quiz, because about every other term I have a student who, for some reason, it doesn't work on their system, or it doesn't work immediately on their system, and you'll need to contact the computer help desk and they can get it up and running on your system. I'm not sure what the problem is, but it's just that it's not configured right on your system. So far, everybody's been able to get it to run, although, like I said, sometimes people have had to contact the computer help desk, or whatever it's called at Yavapai, to get it up and running. Another note, we're going to start Chapter 3 today. We're going to skip Chapter 2 because at Yavapai, you should have covered the material in Chapter 2 in a previous course. However, if you want a refresher, I have Chapter 2 optional, and it's set up the same way. An overview and objectives, a study guide, and then a link to the slideshow. That reminds me, I've now finished Chapter 1 and Chapter 10, so I'm going to make the slideshow available to you. Any questions about that?
I have a question.
Uh-huh.
For the extra credit study schedule, what you really want us to write there? Just the hours that we study?
Yeah, no, just the time that you're studying. So when you're planning to study. So if you're always planning to study from 7 to 8 p.m., you put that on the second tab of the worksheet. On the first tab, you just have your general schedule, what you're doing throughout the day.
Okay.
Okay?
Thank you.
All right. I think that's enough. I don't think there's anything else I need to talk about. This website is pretty much running. I don't have everything available to you, and near the end I will make a change in something, but that's not even available to you yet. Or maybe it is. Yeah, I guess it is, because it's the lecture. I'm going to change chapter 7. Okay. Any questions about anything? If not, let's go to the schedule. So today we're going to be discussing chapter 3 and hopefully ending this lesson. Okay? I've only got it scheduled for one day. There is no lab, but we have a very long day today. So hopefully we'll finish chapter 3. Okay, usually in the chapters I have a preliminary slide giving you the major goals and the rough outline. Now please understand the major goals are not all of the objectives. If you want to see all of the objectives for chapter 3, take a look at the study guide. And at the top of the study guide, I've got the objectives for chapter 3. Okay? All right, any questions? So there's 11 objectives. Boy, I don't have that labeled. Oh, yes, it is. There it is there, objectives. I also give you a rough outline in the first slide, so know the terms, know what is resolution, refraction, know what stains are, know what a basic and acidic stain is, and they're also called dyes. Know how to make a smear preparation, know what a mordant is, and know what a peptidoglycan is. Let me mention that students often get confused between making a bacterial smear on the slide and then a stain of the bacteria. You always make a bacterial smear on a slide before you stain the slide. Is that clear? And you don't always have to stain it. You could look at the cells directly, in which case you'd still make a bacterial smear. All right, be able to understand the sizes of organisms and the properties of microscopes. Understand the major differences between the light microscopes and the electron microscopes. Understand why staining improves resolution, why we use staining. And know that there are 3 types of stains. There's a simple stain, a differential stain, and then special stains. And then lastly, the major outline. Understand the gram stain. We'll talk about it in depth, its procedure, and the basis of its differential staining. Any questions about any of this? All right. For Chapter 3, Observing Microorganisms, we will be using the metric system. It is possible to use the English system, but nobody uses those English units. It's something like a grain is what you'd use to measure microscopic cells, and nobody uses that. We all use the metric system. For the metric system, you need to know that 1 micrometer, also called a micron, is about the size of most bacteria cells. Some of them are larger. In fact, even the average is between 1 and 3 micrometers, but it's about 1 micrometer. You do need to know one size smaller than that, and that is 1 nanometer. Let me see if I can blow this slide up so that the table down here will be readable. Why you need to know a nanometer is because that's the size we measure in an electron microscope. So you should know a nanometer. You don't really need to know a picometer. Boy, that's not showing too well. A picometer is 1 over 1 times 10 to the 12th zeros, a picometer. A nanometer is 1 over 1 to the 10th to the 9th zeros. And we're talking about meters here. So a micrometer is 1 over 10 to the 6 meters, or 1/1,000,000th of a meter. A nanometer would be 1/1,000,000,000th of a meter. You should know a millimeter. That's 1 over 1,000 meters, or 1 times 10 to the 3. You should also know a centimeter, 1 over 100th meter. You don't need to know a decimeter. You should know a meter, and just for equivalence, a meter is just a little longer than a yard, so about a yard. And then you should know a kilometer. That is 1,000 meters. So you should know all of these metric units, and then you should be able to convert from one to another. So 1 meter equals 1,000 millimeters. 1 micrometer equals 1 over 10 to the 6 meters, and that would be 1 times 10 to the negative 6 meters. A nanometer, 1 times 10 to the negative 9th meters. So you are expected to know this for the quizzes. You should know the units, and obviously what the prefixes are and what they mean, and the metric equivalent, so that you can convert from one metric unit to another. Now, converting in metrics is easy because they're all based on a unit of 10. Like a kilometer is 1,000 meters. A centimeter is 1/100th of a meter. Generally, I don't convert from, nanometers directly to centimeters. What I'll do is convert from a nanometer to a meter, and then convert from a meter to a centimeter. So I've got everything set up in my head by a meter. But you can do it however you like. Any questions about any of that? The third part of the table, switching from metric to English and English to metric, is hard. You do not need to know how to do that. Okay? Any questions about any of that? You should also know that meters are the unit of length we use in the metric system. We have the same prefixes for weight, or what is that called? When you're in outer space, you don't have weight, it's mass. So the prefixes are used for the weight or the mass, but we switch the base unit to a gram, and then everything is based on that, like a kilogram or a centigram. Any questions about that? For measuring volume, we don't use meters, we use liters, and then everything is based on a liter. There's a kiloliter and a centiliter. Why you need to learn the metric system is illustrated by this graph. This graph is showing you all the countries in the world that do not use the metric system. And if you notice, there are only 3 of them. And the United States is the only big country that doesn't use the metric system. And in reality, we are slowly switching over to use the metric system. Congress, I think it was when Andrew Johnson was president, our 3rd president, passed a law saying that the United States would switch over to using the metric system. And that switch is still continuing today, and as you notice, we haven't gotten very far. But hopefully you notice, like on cars, they have both miles per hour and kilometers per hour. And sometimes on the road signs, they'll give both. So we are slowly switching over. Another reason for learning the metric system is this is the unit of measurement that is used in science and medicine. As I stated, everybody measures cells in the metric system, how big the cell is. Nobody uses the English system. When we're talking about medicine, they give you so many cc's, and that stands for cubic centimeter, which is 1 milliliter of an injection. Any question about any of that? So you need to know the metric system because science and medicine use it. And then most of the world uses it, all the countries in gray. All right. Let's talk about the sizes of some microorganisms. And we're just talking about typical sizes because cells can be of different sizes. Eukaryotic cells, a typical cell is between 10 and 500 micrometers. A prokaryotic cell is about 1/10 the size of that, or between 1 micrometer and 10 micrometers. 10 micrometers is actually much larger than your typical cell that's a prokaryote. Now, as I stated, you can get cells which are much bigger than this and cells which are smaller than this. The smallest prokaryotic cell is about 0.4 micrometers. And the largest eukaryotic cell that you've seen is the yolk of a chicken egg. That is 1 cell. And I don't know how big that is, but it's close to 1 inch long, so that would be about 2.4 centimeters. Of course, if anyone ate an ostrich egg or even a turkey egg, that would be a larger cell. Okay, not many of us have seen the yolk of an ostrich egg. I have had one or two students who have seen the yolk of a turkey egg. I've never even seen a turkey egg. All right. Viruses, in comparison to a prokaryotic cell and a eukaryotic cell, are smaller. They are about 1/10 the size of a prokaryotic cell. The very largest viruses get up to the size in a diameter, as well as their length, to the size of a prokaryotic cell. And of course they can be smaller than 1/10. What would that be, 0.1? This says their diameter is 0.02. But the point is, on average, they're about 1/10 the size of a prokaryotic cell, which is on average about 1/10 the size of a eukaryotic cell. Any question about any of that? You should know what you can see with your naked eye, what you can see with the light microscope, what you can see with the electron microscope. You should also know that a person is bigger than a chicken egg, which is bigger than a frog egg, which is bigger than most eukaryotic cells, which is bigger than prokaryotic cells or bacteria, which are bigger than viruses, which are bigger than large molecules like proteins, which are bigger than small molecules like water, which are bigger than atoms. So you should know the relative rank of things, but you do not need to know the size. That a small molecule is 1 nanometer. Viruses are around 100 nanometers, a little smaller too. And then a bacteria is around 1 micrometer. Okay, you don't need to know that size. Let me point out an error in this chart, which is really surprising because it was made by Pearson, who publishes textbooks. This is incorrect. That shouldn't be 1 micrometer there. That should be 1 millimeter. And I put in a correction right there. All right, any question about this chart? You should know the microscope. We're going to be talking about a compound microscope because that is the microscope we use in the microbiology lab. A compound microscope is one that has 2 or more magnification lenses. And I've never seen one with more than 2, but I suppose they exist. There is a lens that magnifies that we call the ocular lens right here. And then there's a magnification lens that we call the objective lenses. And most microscopes have 4 possible objective lenses, and they rotate. The total magnification of a compound microscope is the magnification of the ocular lens times the magnification of the objective lens. So if we're using the 4x objective lens, the total magnification of a 10x ocular lens would then be 40x total magnification. Any question about any of that? This slide is also telling you that most compound microscopes have 4 objective lenses. Usually they are the 4x objective lens, the 10x objective lens, the 40x objective lens, and the 100x objective lens. Any question about any of that? You should know the parts of the microscope. Now this one doesn't show you many parts, so let's go on to the next slide. So you should know the parts of the microscope that are generally shown here. And I think you need to know all of these parts. Okay? Note that the fine focusing knob is the little one. The big knob is the coarse focusing knob. You should note that the condenser and the iris diaphragm are together. The iris diaphragm on this microscope is right down here, and the condenser is right there. They serve different functions on the microscope. The condenser puts a beam of light from the lamp and then focuses it on the specimen. So it is, in a sense, a lens, but it's not magnifying. All it is doing is focusing a beam of light on the specimen. The iris diaphragm controls how big that beam of light is. The iris diaphragm has a shutter, and you can open it up so that you can get a total cone of light coming through that's as big as the, what do you call that, the specimen or the objective lens if you're using the 4X. And then you can shut that shutter so that you get a smaller and smaller cone of light. Generally, you want the iris diaphragm to be set so that the cone of light coming through is the size of your field of view, the size that you're viewing under the microscope. And as you go up in objective lenses, the field of view will decrease in size. So you open the iris diaphragm when you're at low power, or the low power objective lenses, and then you shut that iris diaphragm as you go up in power with the objective lenses. Any question about any of this? So know these parts of the microscope, you will be tested on them. Not all microscopes are compound microscopes. You can have a simple microscope. A simple microscope only has one lens. In reality, you could call a simple microscope a magnifying glass, because a magnifying glass only has one lens and it magnifies. And that's what a simple microscope does. The microscope that Antony van Leeuwenhoek made were simple microscopes. And here we're looking at it there. And there's his one lens that magnified. Any question about any of that? All right. Resolution is the ability of the lenses to distinguish between 2 points. All light microscopes have a resolving power of 0.2 micrometers, and you can distinguish 2 points as being 2 separate points from each other as long as they are 0.2 micrometers apart from each other. If they get closer than that, you cannot distinguish them as 2 separate points. And the reason for that is the resolution limit of white light is approximately 0.2 micrometers, which is the same as 200 nanometers. And that is based on the limitation of the wavelength of light. Meaning, the reason why you can't distinguish them as 2 separate points is that the wavelength of light is about 200 nanometers, and it can't distinguish anything underneath 200 nanometers. This means that the best compound microscope, light microscope, can only magnify about 2,000 times. Any question about any of that? All right, let's look at some pictures of resolution to get it more in your head. So these two right here can be distinguished under the microscope. This is what they really are, and this is what they look like under the microscope. You can distinguish these two as being two separate points. However, these two you cannot distinguish as two separate points, and then all of these cannot be distinguished as two separate points because they're too close together. You can't distinguish them under the light microscope. Any question about that? Light bends when it moves from one substance to another, assuming that the two substances have a different refractive index, like air and water have a different refractive index, and so it looks like this pencil is bent right there at the interface between the air and the water. And why it looks like the pencil is bent is because the refractive index of water is different than the refractive index of air, and so light bends right there. And this is showing you what an artist thinks it looks like. I think that's actually a better picture, but any question about that? So whenever light travels from one medium to another, it will bend, and we call that refraction. We use oil because oil has the same refractive index as glass, and so when the light is going through a glass slide containing our specimen and then moves into the oil, the light doesn't bend. And that gives us a better resolution. I'll explain why in a few slides. But let's talk a little bit more about refraction, because it's extremely important for life as well as important for understanding a microscope. When a bird is looking at a fish, because of the bending of light, the image the bird sees of the fish is going to be here, or closer to the surface than where the fish really is. And if the bird wants to catch the fish and eat it, it has to take into account the refraction of light right here, so that the fish is in a different location so it can catch it. And birds are actually good at accounting for the refraction of light, and they know that when they see the fish and it looks like the fish is here, it's really down here. Any question about that? All right. Because of the refraction of light, some of the light that comes through the slide and through the specimen, when it hits the air, some of that light is bent, shown in red, and it's not collected by the objective lens. Obviously, some of the light will not be refracted, and it will be collected by the objective lens, but some of it is lost by refraction. And this slide shows it better. So this wavelength is refracted here and actually there too. And the objective lens is collecting that so we can see the specimen. Some of the light is refracted, and it's refracted away from the objective lens. It is not collected. And then some of the light, actually in this case, doesn't ever leave the lens, at least not here. It might leave the lens down here. Okay? We use oil because when we use oil, there's no refraction of light between the glass and the oil and the glass of the objective lens, because oil has the same refractive index as glass. And that way, more of the light, meaning this one, doesn't get bent away from the objective lens, and so it's collected by the objective lens. So the objective lens is catching more of the light coming through the specimen, and so that will give us better resolution. And that's why we need to use oil with the 100x objective lens, because that lens is very small, and so a lot of the light that goes through the specimen is refracted away from the objective lens, and if we don't catch that light, we can't see the specimen. And if more light comes in, the better the resolution. Any question about that? So oil reduces the refraction of light, so more light goes into the objective lens, giving us a better resolution. And that will be a question, I think, on the worksheet. Brightfield illumination is what is used on most microscopes. Light comes from a lamp, shines through the specimen, and we have a light region around the specimen from the light coming from the lamp. And the specimen is lit up, but it'll be darker because of usually the stains that we stain the cell with, but it could be the color of the specimen as well, if it's a colored specimen. Any questions about Brightfield illumination? That's what most microscopes use. The light comes from the lamp and goes through the specimen and goes directly into the objective lens as well as the ocular lens of the microscope. Dark field illumination is a little different. We have a disc that blocks the light directly coming from the lamp and then going directly through the specimen and directly to the microscope. Because the light is blocked, the background of the slide looks dark because there's no light coming from the lamp. It's blocked by the disc. The specimen lights up only because some of the light coming through from the lamp to the specimen gets refracted into the objective lens. So only the light that its wavelength is changed by the specimen so that the wavelength is directed towards the objective lens is captured by the objective lens. And so the specimen will show up lighted, meaning it's colored. It will be a little darker than the Brightfield illumination, but it's essentially the same as the Brightfield illumination. And then the background is dark because no light is directly coming from the lamp. Any questions about Brightfield illumination and dark field illumination?
I got a question.
Uh-huh.
So what would be like an example of why someone would want to use one type of illumination over the other?
Um, well, if the specimen isn't stained very well, it's not going to show up very well under bright field illumination. It'll show up better in dark field illumination. So that would be one reason why you would use it. It's just a different way of looking at the cell, and you can sort of see that it does look different. For example, you can more easily see the vacuoles, like that one right there and that one right there, on this specimen than compared to this specimen. All right, so you will be tested on bright field illumination. Dark field illumination, you only will be tested on the concept. I will not be asking you direct questions about dark field illumination. Okay? Here is an electron microscope. They do not use light to see the object. They use a beam of electrons instead. So electron microscope uses a beam of electrons to view the specimen. And an electron has a much smaller wavelength than visible light. So it can give greater resolution and you can use greater magnification. And the beam of electrons are focused on an electromagnetic lens. It's not glass. It allows examination of much smaller specimens as well as details to specimens we would not see under the light microscope. For example, we can use the electron microscope to view viruses. Viruses are generally too small to be seen in the light microscope. Any questions about any of that? We can also use the electron microscope to see internal cell structures, like the different organelles in this cell here. There are two main types of an electron microscope. Obviously, there are other types, and we'll briefly discuss them, but you need to know the two main types. There is a transmission electron microscope, or TEM, right here, and then a scanning electron microscope, shown here, and that would be abbreviated SEM. If you notice, these look different. What do you notice that looks different between these two electron microscopes?
It looks like you can see the inside of structures more on one than the other.
Yes, actually, where do I put that? Maybe down there. On the transmission electron microscope, let's put it up there, you can see, don't ask me why that runs in the middle of the lecture. You can see structures in the transmission electron microscope. You do not see structures generally in the scanning electron microscope. At least you do not see structures inside the cell of a scanning electron microscope. What else do you notice that's different between this picture and that picture? Okay.
A lot more
Which one?
detail.
Yeah, we've already talked about the details. Which picture looks two-dimensional? Which picture looks three-dimensional? Okay, can you see the bottom of this cell? Or the top of this cell? You guys don't like to talk, huh? All right, we'll move on.
The one on the right seems a little bit more 3D.
Yeah, the one on the right is three-dimensional. We're only scanning the surface in scanning electron microscope. Transmission electron microscope is seeing a plane through the cell. So all we're seeing is a two-dimensional image that is only a plane, meaning, what do you call that, one layer thick through the cell. Not that. What do I want to do? There are other types of electron microscopes, and we'll talk about two other types in just a minute. Here's another scanning electron microscope, and you can see it's in 3 dimensions. And this is some crystal. This is a transmission electron microscope looking at the section of what is it, a chloroplast, and you can see the different layers of something in the chloroplast. Okay, but they're not 3 dimensions, and you can see, I don't know, structures within the cell. You will not be tested on this, but I just want to show it to you. There's also a scanning tunneling microscope showing you 1/100th of an atom in a protein of E. coli. It uses a metal probe to scan the specimen. There's also atomic force microscopy, a 4th type of electron microscope. It also uses a metal and a diamond probe inserted into the specimen. It produces 3-dimensional images of the object. And these are very small. 10 nanometers, 2 nanometers. All right, any questions about the electron microscope? You do need to know the 2 major ones, the transmission electron microscope and the scanning electron microscope. Let's talk a little bit about making preparations of a specimen to view under the microscope, and then staining. Which of these cells, the ones on the left or the ones on the right, are easier to see?
The ones on the right.
The ones on the right. The only difference between these cells and these cells is that these cells have been stained. And because they are stained, they are much easier to see. The purpose of staining is just to increase the contrast between the background of the slide and the specimen. It's all staining, well, I should say simple staining, is intended to do. Increase the contrast between the background and the specimen. Why we need to do that is because cells are not very visible. They have about the same contrast, the same coloration as the background of a slide. And that's because they're mostly water. Light can readily go through them, and so they look transparent, like that one there, or at most translucent. And here the cells look opaque, the dark ones. Okay? So most microbes are colorless, and they have little contrast with the slide or the specimen. Now, obviously, if you're looking at a photosynthetic cell, it would have pigments in it to collect light, and those would be colored. But most microbes are colorless, and to see them well, we need a stain, also called a dye, to stain them, to color them. So staining colors the specimen, or it colors the background to increase the visibility of the specimen and increase the contrast between the specimen and the background of the slide. Before we stain the specimen, we always make a smear. And if it's a bacterial smear, we call it a bacteria smear, a thin film of solution of microbes on a slide. And when students make a bacterial smear, the number one error they use is to use too many cells. You only want a few cells so that when you spread them out, they are ideally only one layer thick. So you want a thin film of cells, and then that thin film of cells will stain well. So prior to staining, the microbes are put in a smear. They're usually fixed to the slide. That's to attach them, and it does tend to kill the cells as well. And we usually do this, make a bacterial smear, before we stain the cells, before we stain the slide or stain the cells. Any question about any of that? Stains, also called dyes, are salts consisting of a positive and a negative ion. If you have one, you usually have the other. So most stains have both a positive ion and a negative ion. If the positive ion is the one that is colored and doing the staining, the colored part we call the chromophore, and if it is the positive ion, then we call it a basic dye. The chromophore is in the cation, and a cation is the one with the positive charge. If, on the other hand, the colored portion of the stain is in the anion, the negative part of the salt, then we call it an acidic dye. Any questions about that? Most dyes in microbiology are basic dyes, but we do have some which are acidic dyes, and usually we call that an acidic dye. We call the stain a negative stain. You'll see why we call it a negative stain in just a minute. Bacteria have a slightly negative charge. So bacteria have a slightly negative charge. Therefore, the basic dye stains the cells a little better than an acidic dye because the positive chromophore is attracted to the negatively stained cells. And that will cause the cell to stain the color of the dye. Some basic dyes include crystal violet, methylene blue, and these two are the most commonly used, but also malachite green and safranin. Any questions about basic dyes and then the difference between a basic dye and an acidic dye? An acidic dye does not stain the cell. It typically stains the background, but not the microbe. Some examples of an acidic dye are eosin, acid fuchsin, and nigrosin. When we stain the background instead of the cell, we call that a negative stain. Let's look at the difference. These cells are stained with the basic dye, and the cells stain darkly. These cells are stained by a negative stain, and the background is stained dark. The cells are light because they are not stained. Both ways, we can easily see the cells. Any question about that? All right, so both of these are a simple stain. A simple stain is where we use one dye to stain the cells. And let's talk about the different types of stains. Did I go the wrong way? Yep. All right, there's three main types of stains in microbiology. There's a simple stain, there's a differential stain, and there's a special stain. As I stated, the simple stain is using a single dye, and it can be a basic dye. I should change that. It's usually a basic dye, but it can be an acidic dye. If it's only one stain, it's a simple stain. The stain can be in an aqueous or alcohol solution and then applied to the bacterial smear to stain the cells. The primary purpose of the stain is just to increase the contrast between the background of the slide and the specimen. The stain is applied to a fixed bacterial smear for a certain length of time. It's then washed off, usually dried and examined. Any question about any of that? A simple stain may occasionally use a mordant. A mordant is something that intensifies the stain. So the mordant is not a stain. It simply intensifies the first stain. The mordant may increase the affinity between the stain and the specimen. Like we'll talk about the Gram stain. The mordant in the Gram stain is iodine, and it simply binds to the first stain and intensifies the staining. You can also use a mordant which simply coats the specimen, putting on another layer, therefore making it much easier to see the specimen. And flagella staining is using a mordant to coat the first stain, and in reality with the flagella stain you're using the mordant repeatedly, so it gets more than one coat. Any questions about a simple stain or a mordant? All right. We're now at 2 o'clock. Would you like to break for half an hour or continue going and then end the class a half an hour early?
I would prefer to end early.
Me too.
Okay. Anyone else?
Same here.
All right. So, what time are we going to stop? Does anyone know? If we stop a half an hour early?
3?
315. So when it gets near 315, please remind me. Okay? And I'll try and keep an eye on it. Boy, we've got another hour and 15 minutes to go.
This might sound stupid, but could you give us an example of like what substance a mordant would be in case I missed?
I'll give you an example of a mordant when we're talking about the gram stain. And we'll talk about that in a few slides. Will that be okay?
Yeah.
Okay. Like I said, in the gram stain, the mordant is iodine, and I'll talk more about that with the gram stain. Usually, when we're working in microbiology and using a simple stain, we don't use a mordant. However, with the flagella stain, which is not a simple stain, it's a special stain, we are using the mordant repeatedly. All right, a differential stain is a stain that reacts differently with different kinds of microbes. This allows you to distinguish between two types of microbes. A differential stain usually uses two or more stains, but that is not absolutely required. If one stain, and we use a treatment, meaning a special treatment of that one stain, if it does stain the cells, two different species of cells differentially, then that would be a differential stain. But usually a differential stain has two or more dyes. The most commonly used differential stains in microbiology are the gram stain and the acid-fast stain. And both of these stains use two stains, meaning two dyes. Any question about that? All right, the gram stain was invented by Hans Christian Gram in 1884. You do not need to know the year, but you should know that the gram stain was invented by Dr. Gram. And so whenever you write it, you should use a capital G with the gram stain because it is named after somebody. And we always capitalize somebody's name. Any questions about that? Now, Hans Christian Gram invented the gram stain so that he could distinguish between the cells of a patient and the cells of a bacterial infection, meaning his differential stain stained the human cells one color, which was red, and the bacterial cells a different color, which was purple. And so he did intend it to be a differential stain. It's just that he didn't realize how differential it was. And microbiologists started using his gram stain, and then they discovered that it was a very useful stain in bacteriology because the gram stain classified almost all bacteria into two very large groups. The gram-positive cells, which stain dark or purple, and the gram-negative cells, which stain reddish or pinkish, or kind of, this looks like a reddish purple. Okay?
Is it the negative that have the thin wall or the thin wall?
Okay, can you get a little closer to your computer? I didn't hear that.
Does the gram-negative stain, the pink ones have a thinner cell wall?
Yes, I'll be talking about that. The gram-negative cells have a thinner cell wall than the gram-positive cells. we'll talk about that when we're talking about the gram stain in a little bit. Okay? But that'll be several slides in the future. Anyways, the gram stain proved to be very useful because it divides almost all bacteria into two categories of cells. Before we stain any cells, remember we make a thin bacterial smear on the glass slide. And I'm surprised I don't have how we're making the bacterial smear on here. And then you allow that to air dry. You then heat fix the bacterial smear, and the air drying and the heat fixing are to attach the cells to the glass of the slide. Otherwise, in the water washing, the bacteria will wash off. Another reason for air drying and heat fixing the cells is that tends to kill most bacteria. Obviously, it doesn't kill endospores, but it does kill most bacteria. All right? So you make the bacterial smear, you air dry, you heat fix. You then take that slide and treat it with the gram stain. So in reality, only step 3 to step 10 is part of the gram stain. Before that, that's the treatment of the bacterial smear, and then 11 is actually just viewing it. Okay? Any question about that? So to start the gram stain, you take a bacterial smear and you flood it with crystal violet. You apply the stain for 60 seconds, meaning you leave the stain on the cells for 60 seconds. You then dump that off and wash gently with cold water. That's to remove the excess crystal violet. And then you remove the excess water. You don't blot it or anything, you just tip it and get the water off. You then flood the slide with iodine, and iodine is a mordant. You apply the iodine for 60 seconds. What the iodine is doing is it's just complexing with the crystal violet, making a larger molecule, and that will intensify the color of the crystal violet. So iodine is not a stain here, it is a mordant. Any questions about that? You could use iodine as a stain if you were staining starch. Like if you're staining potato cells of a potato and you use iodine, you will stain the cells and the iodine will stain wherever there's starch. Any questions about that? But in the gram stain, we're not using it as a stain, we're using it as a mordant. You then dump the iodine off and then wash the slide gently with cold water. You then dump the water off and then you flood the slide with acetone ethanol mixture, or it could be something else. We'll just use this because that's what's in the slide. And this is the decolorizing agent. You apply the decolorizing agent for a short period of time, about 5 seconds. That doesn't show up. Let me see something that'll show up. Here, maybe this will show up. Not very well. Let's get it over my face. I'm not sure why my face shows up. So many other things don't. When you're applying the decolorizing agent, it's best to apply it at the top of the slide and then let it run down the slide. And when the crystal violet stops running on the slide, you stop applying the decolorizing agent. That's the best way to apply the decolorizing agent, meaning you apply it only for the time you need. Once the crystal violet stops running, you stop applying the decolorizing agent. Any question about that?
I have a question.
Uh-huh.
So you're applying that decolorizing agent just to remove extra dye that's going to make it so you can't see things clearly? I don't really understand why you're colorizing if you're trying to stain something.
I'll talk about that in a little bit. At the moment, I'm just going through a brief overview, but you're decolorizing to remove crystal violet from the cells that do not retain crystal violet. If the cells retain the crystal violet, you do not remove the crystal violet. And obviously, the background of the slide will not retain the crystal violet, and you're removing the crystal violet from the background of the slide. You then wash immediately with cold water. Why this washing step needs to be immediate is if you leave the decolorizing agent on the cells, it'll continue acting. And it can pull crystal violet out of the cells that normally retain the crystal violet. Meaning if you leave the decolorizing agent on too long, it will remove crystal violet from cells it should not remove the crystal violet. Does that make sense? So the decolorizing agent is applied only for a short period of time, and then you wash it out. So the decolorizing agent is totally removed from the cells. You then invert the slide to get off the majority of the water, and then flood the stain or the slide with safranin for 1 to 2 minutes. When you're done, you pour off the safranin and then wash it gently with cold water. And that's the end of the gram stain. After that, you can blot the slide dry. We'll talk about blotting in a little bit. You get bibulous paper. Let's see if it does show up. You get bibulous paper, put the slide inside the bibulous paper, and then blot it. You do not rub the slide against the bibulous paper, because if you rub it, can't get it to show up now. There we go. If you rub it, it will remove the cells. So you just blot it. Blotting removes enough of the water so that you can then view the slide under oil on the electron microscope. The small molecules of water that are remaining on the slide will not interfere with the oil. Now you'll notice that in my procedure for doing the gram stain, everything is 1 minute of application except for the applying the decolorizing agent. Okay? And when you're writing the steps of the gram stain, you do not have to put in flood with crystal violet, wash, flood with iodine, wash. All you need to do is state at the start or state at the end. After each step in the gram stain, I wash with cold water. Okay? Any question about the procedure of the gram stain? Not the specifics of the gram stain, just the procedure. All right, let's look at the gram stain in a little more detail. When we stain different cells with crystal violet, they all take up the crystal violet and they all look purple. Any question about that? So even the gram-negative cells at this point look purple because they take up the crystal violet. We then apply the iodine. Don't ask me why it's red, because iodine is actually brown. And it doesn't change much the color of the cells. They will be a darker purple. So instead of a blue purple, they'll be a purple purple, if you know what I mean. And they may be more intense. So if this one is lightly stained, it might be darkly stained here. Okay? Any question about that? The point is, both the gram-negative and the gram-positive are purple after applying the iodine. We then water wash, remove the water, and then apply the decolorizing agent, which here is an alcohol wash. The alcohol wash, or the decolorizing agent, removes the crystal violet from the gram-negative cells, but does not remove the crystal violet from the gram-positive cells. The gram-positive cells then stain purple after the alcohol wash. The gram-negative cells stain clear after the alcohol wash. Any question about that? So you can see at this step, the gram stain has already differentiated the gram-negative cells from the gram-positive cells. So this is the differential step in the gram stain, the decolorizing step. Any questions about any of that? All right. We then apply safranin, which will stain the gram-negative cells pinkish or red because they were clear. And we apply safranin because clear cells will not show up very easily or very well under a light microscope. All right. Any questions? Well, here's a question for you. Why doesn't the safranin get in the gram-positive cells and stain them at least a little bit pinkish? Anybody have a guess? We're applying the safranin here. Why doesn't it stain the gram-positive cells at least a little bit pinkish?
Is it just because the color's darker in the purple, so you're not going to see it?
I think you said that right. I didn't hear all of it, but it sounded like you were saying the purple is more intense of a stain, and our human eyes only sees the purple. If we were to look at the gram-positive cell under the electron microscope, we could see safranin in portions of that cell, and then we would see crystal violet as well in portions of that cell. And to our eyes, the purple is more intense. Our eyes only see the purple, but the safranin is there in the gram-positive cells. To our eyes, though, we just don't see it. Like I said, to see it, we would have to view the cell under an electron microscopic image, and that would be a TEM, transmission electron microscopic image. All right, any questions about any of that? So let's talk a little bit more about applying the crystal violet. Crystal violet sticks to peptidoglycan in the bacterial cell wall. And that's why the cells stain purple. Crystal violet is considered the primary stain because it's applied first and it colors all of the cells purple. Meaning it colors both the gram-negative and the gram-positive. So after applying crystal violet, the gram-positive cells are purple in color. The gram-negative cells are also purple in color. We then apply iodine as a mordant. It increases the affinity of the crystal violet for the specimen. The iodine is actually complexing with the crystal violet in the specimen. After the iodine step, the color of the gram-positive cells is purple. The color of the gram-negative cells is purple. We then treat the cells with the decolorizing agent. The decolorizing agent might be acetone and ethanol. You apply the decolorizing agent for only about 5 seconds. It's best to apply it until the crystal violet stops running, and then you wash immediately with cold water to stop the decolorizing agent from acting on the cells. The decolorizing agent washes the crystal violet out of the gram-negative cells, but it does not wash the crystal violet out of the gram-positive cells. Any question about that? As I already mentioned, the decolorizing step is the differential step in the gram stain. Because after the decolorizing step, the cells look different. The gram-negative cells will be clear, the gram-positive cells will be staining purple. All right, let me note that when you're applying the decolorizing agent, if you apply it for too long, it will remove crystal violet iodine complex out of the gram-positive cells. That's why you decolorize only for about 5 seconds. Of course, if you don't use the decolorizing agent long enough, then you will not remove crystal violet out of the gram-negative cells. So the application of the decolorizing agent is very tricky. Okay? Any question about any of that? So the gram-positive cells after decolorizing are stained purple, and the gram-negative cells are colorless, are not stained, because the crystal violet was pulled out of the cells by the decolorizing agent. We then apply safranin. It's a pink dye. It does stain all bacteria. It's called a counter stain because it has a different color to the primary stain, the crystal violet, and it's also applied as a secondary stain. After applying safranin, the color of the gram-positive cells will be purple, at least to our human eyes, and the color of the gram-negative cells will be pink. Any question about that? You then blot the slide with bibulous paper, and then you can examine it under the 100x objective using oil. Of course, it doesn't need to be viewed immediately, but if you did want to do that, you'd want to blot it dry. What is the basis of the differential staining? Why do gram-positive cells stain purple and gram-negative cells stain pink? It has to do with the relationship of the cell to its cell wall. The gram-positive cells, gram-positive bacteria, have a thick peptidoglycan. And we'll talk about peptidoglycan in our next lesson. But what it is a disaccharide molecule with some amino acids. Anyways, the gram-positive layer have a thick layer of peptidoglycan, and it's just layered on top of each other in a very thick layer. The gram-negative cells, on the other hand, have a very thin peptidoglycan layer in their cell wall. It does contain several layers of peptidoglycan, all layered on top of each other, but compared to the gram-positive, that layer is very thin. The gram-negative cells also have a lipopolysaccharide layer as part of the outer membrane of their cell wall, and we'll talk a little bit more about that in our next slide. So here we're looking at a gram-positive cell, and as you can see, the purple part stands for the cell wall. It's very thick. The yellow part is the cell membrane underneath. And here, let me blow that up a little bit. Here we're looking at the gram-positive cell wall, and you can see that it has layer upon layer of peptidoglycan. Now this artist chose to represent it by having 3 layers of peptidoglycan. In reality, this is hundreds of layers of peptidoglycan. It's not 3, it's hundreds of layers. And this would be the cell membrane underneath the cell wall. Okay? So the gram-positive cells have a very thick cell wall, and the crystal violet cannot wash out of it. On the other hand, the gram-negative cells have a much thinner layer of peptidoglycan in their cell wall. And you could say they have a thinner cell wall. It's actually a thinner peptidoglycan layer. Outside of the peptidoglycan, the gram-negative cells have a lipopolysaccharide, which is part of the cell wall. Let me blow that up if I can. And you'll notice that it is a lipid bilayer, the same as the cell membrane. And in fact, the lower layer of the bilayer is exactly the same as the cell membrane. It's mostly made up of phospholipids. It does have some proteins in it, but it's mostly phospholipids. There's a protein right there. The outer layer of the bilayer of the outer membrane of the cell wall does contain phospholipids, like there's one right there, and all of these yellow are a phospholipid. But it has another molecule, and that's the lipopolysaccharide. A lipopolysaccharide is a lipid attached to a polysaccharide, and with this lipopolysaccharide we have the lipid A, which is the round portion in blue, and then we have an O polysaccharide coming out of the lipid A, and that's the hairs in this picture. Okay? Any question about that? The point is that this is the cell wall, and it has a thin peptidoglycan layer. The artist here has shown it is only one layer of peptidoglycan. That is not correct. It's several layers of peptidoglycan, one on top of the other, but it is a thin layer of peptidoglycan compared to the thick layer of the gram-positive cells. And then on top of the peptidoglycan, we have the outer membrane of the cell wall, and it contains lipid A in it. And lipid A is important because it is an endotoxin. We'll talk about toxins later in the term, but for now, let's just mention that a toxin is something that is toxic to us, and if you get enough of them, they will kill you because they're toxic to us. And lipid A is an endotoxin. Any questions about any of that? So, the crystal violet iodine complex forms in all cells, both the gram-positive and the gram-negative. But in the gram-positive cells, the crystal violet iodine cannot be washed out of the thick peptidoglycan layer. As long as that layer is intact and thick with the normal procedures of the gram stain, the crystal violet iodine will not wash out of these cells, and therefore they remain purple. In the gram-negative cells, the crystal violet iodine is washed out because the peptidoglycan layer is thin, and the lipopolysaccharide layer will be disrupted by the alcohol in the alcohol wash. Actually, if we're using acetone, acetone will also disrupt the lipopolysaccharide layer. Therefore, the crystal violet iodine washes out, and so the cells will appear colorless. Any question about any of that? Any question about why the cells stain differentially? All right, no questions, let's move on. Here we're looking at a slide which is staining with the gram stain, and you can see gram-negative rods there, gram-negative cocci there, and a whole bunch of them here. Did I say that right? Gram-positive rods and gram-positive cocci. Here is a gram-negative rod. Where's a few more? Oh, that's probably one there. Oh, there's two there, here and here. Gram-negative rods. And then a gram-negative vibrio. And there's another one. And then a gram-negative spirilla right there, and a whole bunch of them here. I don't see any gram-negative cocci, but that is an unusual cell. All right, any questions about the gram stain? Separating cells into two categories, the gram-positive cells and the gram-negative cells. Now, when you're doing the gram stain, you will have complications or problems. Like this is actually very similar or very commonly seen in the gram stain. And how do you view that? Are these gram-negative, and are these gram-positive? When you're looking at a slide like this, you should understand that these are bad regions of the slide because there's a crystal of crystal violet that has formed on the slide. Probably what happened was the crystal crystallized in the bottom of the crystal violet, and then when the person put the crystal violet on this slide, the crystal came out, and there's the remainder of the crystal on the slide. Sometimes around the crystal violet crystal, you can get dark staining of the cells. So the cells further out stain gram-negative, but near the crystal they stain gram-positive. And this is considered a bad region of the slide. This is considered a bad region. These have no crystals in them, so these are good regions of the slide. When you're using a slide, only look at the good regions of the slide. Don't record your observations in a bad region of the slide, because the color could be thrown off by the crystal. Anyways, with the good regions of the slide, the cells stain well, and it's easy to distinguish whether they're gram-positive or gram-negative, as well as the cellular growth arrangement. Like this looks like these cells tend to grow in streptococci. All right, any questions about that? Now the gram stain has some complications or problems, and if you were to use it, you may pull your hair out because it is tricky, especially applying the decolorizing agent for 5 seconds or less. It's tricky. Most consistent results are obtained with the gram stain when it is used on young, growing bacteria. And when I say young, I mean cells that have been growing for 2 days or less. If the cells are growing for more than 2 days, the gram stain may not stain the cells correctly. The gram stain is not universally applicable. Some bacteria cells stain poorly if they stain at all. The cells that stain poorly are the Mycobacteria, and I've already talked about these, I think it was yesterday in our lesson. The Mycobacteria have mycolic acid in the cell wall, and mycolic acid is a lipid, and the gram stain is water soluble, so the lipids do not stain. I didn't say that right. The lipids prevent the stain from getting into the cell. Any questions about any of that? Usually the Mycobacteria will stain at least lightly if you use it on young cells. Okay? And if the decolorizing step was not done correctly, like if it was not done long enough, some of the gram-negative cells will stain gram-positive, meaning purple, because the decolorizing agent did not remove all of the crystal violet. And if it's not, or if it's applied too long, the decolorizing agent will remove the crystal violet iodine complex from the gram-positive cells, and then when you apply safranin, those cells will stain pinkish, even though they're gram-positive. So the decolorizing step is really tricky, and the gram stain is tricky. Any questions about that? When students have used Mycobacteria or another bacillus species, meaning a species of bacteria that forms bacilli, and they use it on old cells. I've occasionally seen that the slide is staining the same cells, some of them gram-positive and some of them gram-negative. And when I asked about it, the cells were growing for 2 weeks or more. And I said, well, that's why it's not, the gram stain isn't working, and that is your cells are too old. That is particularly true when you're working with the mycobacteria. Okay, so you need cells that are young, that have been growing for less than 2 days or less. Otherwise, the stain, the gram stain, may not work correctly. Do I have a picture of that? I do not. What's happening with the old cells is that some of them die and the cell wall starts deteriorating. When that happens, the crystal violet can wash out of the gram-positive cells because their cell wall is decaying. And that's why you don't want to use old cells, because old cells will have lots of decayed cells. Any question about any of that? All right. If not, let's talk a little bit more about gram-positive and gram-negative cells. The structure of the gram-positive and the gram-negative cell walls not only affects how they stain in the gram stain. The structure of the cell wall is also related to how they will respond to certain antibiotics and other treatments. Gram-positive bacteria tend to be killed easily by penicillin G. Any questions about that? And they're killed easily by cephalosporin and many detergents. The gram-negative bacteria are more resistant to these antimicrobial treatment. Let's talk specifically about penicillin G. Let me go back. Here we go. I guess I do it down here. Yep, too far. Why did I lose where I was? There we go. The gram-negative cells have an outer membrane on the cell wall. This outer membrane stops water-soluble molecules from coming through it. If something comes through it that's water-soluble, it probably comes through the porin protein, and I don't want to talk about that now. We'll talk about that in our next lesson. The point is, this outer membrane can stop penicillin G from getting through the outer membrane and then doing damage to the gram-negative cells. So penicillin G cannot work on gram-negative cells. Any question about that? And then in the gram-positive cells, the penicillin G can get to the cells and do the damage. So penicillin G can work on gram-positive cells, but not on gram-negative cells. Any question about that? All right, there's no questions. Let's mention that this outer membrane can protect the gram-negative cell from other treatments, like cephalosporin and some detergents. Some soaps and detergents will be stopped by this outer membrane, and they will not do damage to the gram-negative cell. Any questions about any of that? So the structure of the cell wall has more than just determine how the cells will stain with the Gram stain. It also determines how easily the cell can be killed by certain agents like penicillin G, cephalosporin, and many detergents. Okay? Now, you might ask, how do we kill a gram-negative cell? Well, there are antibiotics which we can use against gram-negative cells, like the, oh, I'm losing it, sorry. I can't remember that antibiotic now. I had it on the tip of my tongue, and then I've lost it, and I can't remember. There's an antibiotic which gets through the outer membrane of the cell wall because the antibiotic is soluble in lipids. So it gets right through the outer membrane of the gram-negative cell. And it's a really streptomycin. That's the antibiotic I was trying to think of. It can be used on gram-negative cells, and it gets right through the outer membrane and can kill the cells. But that's not all. Ampicillin, which I think that was about the third member of the penicillin family that was developed. So it's an early penicillin, meaning an early member of the penicillin family. Ampicillin can get through the outer membrane of the gram-negative cell. So ampicillin can kill both gram-positive cells and gram-negative cells. And we have a lot of antibiotics that can work on both gram-positive and gram-negative cells. But penicillin G cannot work on a gram-negative cell. Any questions about any of that? If not, let me talk briefly about special stains. And I've got it set up that it's not examinable, but I need to talk about it because you may need to do a special stain when you're working on the unknown project. So some of you may need to apply the capsule stain, which is a special stain. And it's a stain that's used for staining or looking for the cell capsule. Some cells make a capsule around them, and this is actually a differential stain, this one, because we're staining both the cells, darkly stained, and the background of the slide. I'm sorry, my phone's ringing, I'm not going to answer it. The capsule does not stain in the capsule cell stain, and so it appears clear. Any questions about that? And here is a capsule stain that you may use in the unknown project. It's staining with India ink. India ink is a negative stain. It stains the background dark. It also lightly stains the cell, so you can see the cells very easily. They are lightly stained, not as dark as the background, but it does not stain the capsule around the cells. And this is a special stain and a negative stain. All right, any questions about, well, staining in general, the gram stain, or the capsule stain? All right, if there's no questions, this is the end of the lesson, so I'll end it here, and I will see you next Monday. let me remind you that for the infectious disease project, you need to pick a topic and send it to me in an email. You should pick an aspect of the topic, because I will allow 2 people to work on the same infectious disease as long as one person is taking one aspect of it and another person is taking another aspect. You cannot work on the same aspect of the same disease. All right? And the first person who gets it in will get that topic. Please send it to my Canvas email website. I will check that every day, Monday through Friday, and I'll check it about every other day, Friday through Sunday. The instructor email at Yavapai, whatever that is, I will check that, maybe twice or 3 times a week. Okay? I will not check that every day. All right. If there's no questions, I'm going to end it here, and I'll see you on Monday. All right.